I'm Tanubi. I'm practicing at JSL College, Mysore. Today I'm here to present a seminar on drug development and analysis. Let us learn about how to develop a drug and analyze the drug from a compound. The content of these seminars are like this: introduction, design, chemical synthesis, primary assay in vitro, secondary assay to check metabolism in vivo, SAR-led and lead compounds, characterization, ligand compound, clinical candidate, and lastly conclusion. Now let us move on to the introduction of how to develop and analyze the drug. The drug research and development process is a complex and multi-step journey that transforms a potential therapeutic compound into a safe and effective medicine. It involves multiple stages including discovery, preclinical testing and clinical trials, and regulatory approvals. Analysis plays a critical role in each stage, ensuring the quality, safety and efficacy of the drug. As we know, the drug research and development process is not as easy as we think. So, it is a complex and multi-step journey that transforms a compound into a safe and effective medicine. As the patient consumes it, it should be safe. It involves a multiple stages such as the discovery, preclinical testing, clinical trials, and regulatory approvals. Here, analysis plays a critical role in each step, such as maintaining the quality of the drug and the safety. It should be, we should know that it is safe to consume, and also the efficiency of the drug should also be known. Here, it is the flow chart of the drug research and development process. Let us know one by one in, in this seminar. Stage 1 is design. The journey begins with design, where researchers identify potential therapeutic targets and design molecules to interact with them. This stage involves understanding the underlying biology of the disease and using computational tools to design molecules with desired properties. The design stage is critical as it sets the foundation for the entire drug development process. Here, the drug development process starts from the stage that is design. Here, the researchers identify potential therapeutic targets from a compound, and then it is designed to interact with the diseases. And as we know, the design stage is a critical stage in an entire drug development process. Stage 2 is chemical synthesis. The designed molecules are then synthesized in the laboratory through chemical synthesis. This stage involves developing efficient and cost-effective methods to produce the desired compounds. Chemical synthesis is a crucial step as it enables the production of large quantities of the compound for further testing. The designed molecules are then synthesized in lab for further simplifications. This stage involves developing efficient and cost-effective methods to produce desired compounds. Next is stage 3, which is primary assay in vitro. The synthesized compounds are then tested in primary assay in vitro, which involves laboratory tests to assess their efficacy and safety. These assays help identify promising compounds that can be further developed. Primary assays are typically high throughput screens that allow researchers to quickly test large numbers of compounds. Stage 4 is secondary assay to check metabolism in vivo. Compounds that pass the primary assay are then tested in secondary assay to check metabolism in vivo, which involve animal studies to assess their metabolism and pharmacokinetics. This stage helps identify compounds that are likely to be safe and effective in humans. Secondary assay provides more detailed information about the compound's behavior in vivo. Stage 5 is SAR and lead complexes. The data from the secondary assay are used to identify SAR, which is also known as specific absorption rate, and lead compounds. Specific absorption rate analysis helps researchers to understand how the structure of the compounds affects its activity, and lead compounds are identified as potential candidates for further development. Specific absorption rate analysis is a critical step as it enables researchers to optimize the compound's structure for improved efficacy and safety. Next is stage 6, which is characterization of ligand complexes. The red compounds are then characterized in more detail through characterization ligand complex, which involves studying their binding properties and interaction with the target protein. This stage provides valuable insights into the compound's mechanism of actions and helps identify potential issues with specificity and selectivity. Next and last stage is clinical candidate. The final stage is the identification of a clinical candidate, which is a compound that has shown promises in preclinical studies and is ready to tested in humans. The clinical candidate is selected based on their efficacy, safety and pharmacokinetic profile. Throughout the drug development process, a collection of compound is maintained, which involves storing and managing the various compounds synthesized and tested during the process. Last is conclusion. The drug research and development process is a complex and multi-step journey that requires careful planning, execution and collaboration among various stakeholders. By understanding the different stages involved in this process, we can appreciate the challenges and opportunities that come with bringing new medicines to market. The flowchart provided is illustrated the various stages involved in the drug development process from design to clinical candidate. Drug development and analysis is a complex and multidisciplinary field which involves the expertise in chemistry, biology and pharmaceutical field. By understanding this, we should work towards the safer and most effective treatment for patients. Uh, Thank you.